Welcome to Better Biopharma. I'm your host, Tyler Minichello, and today we are joined by Dr. Richard Yu, the CEO and co-founder of Abalone Bio, a preclinical stage biotech that uses AI for functional antibody discovery. Richard's background is in biophysics and computer science, which gave way to his work in protein engineering, structural biology, systems biology, and synthetic biology. Richard, thank you so much for being here.
Hey, Tyler, thanks for having me. Yeah, lots of ologies. I gotta figure out a way to shorten that, but
A lot of ologies. We can all just be half as lucky. We can
yeah,
have so
no,
many ologies
yeah.
after our name one day.
Beatles Road, long and winding road. All about that. Yeah, okay.
So Richard, when I was looking into Abalone Bio, the first thing that I saw was about your fast functional antibody selection technology
Yeah,
platform. And so I wanted to start off with maybe you explaining to the folks at home what exactly fast is, and we'll start from there.
yeah, yeah. Maybe take even just one further step back just to set it up properly, and the reason why we even care about antibodies in the first place, right? So for those in the audience who are not familiar or super familiar with the drug space, starting at the sort of how to explain this to grandma at the Thanksgiving dinner table level. Antibodies, of course, are nature's sort of bouncers, right, in the in the body. They're really good at selectively identifying troublemakers like bacteria and viruses, and taking them to the back door and getting them out of your body or otherwise dealing with them, right? Biotech has turned antibodies into some of the best-selling drugs of all time, right? Humira is one of them, of course, Keytruda. There are just tons of antibody drugs, typically occupying more than half of the top 10 seller lists at any given year. Um, but one of the sort of gaps in antibody drugs has been the ability to either find or design antibody drugs that go beyond that sort of bouncer like, shutting down or inhibition function, right? So wouldn't it be cool if we could take advantages of all the awesome properties of antibodies, like the fact that they last a long time in your body and they're generally safely processed by your body. They're a protein that it's already made by your body. So it's not like some weird small molecule that may get metabolized into some toxic byproduct. Um, and the fact that they're super, super specific, they're these large and chemically complex molecules, they can really fit into and sort of match the surface area, the shape of the different molecules, and really distinguish between different ones quite well. If only we could make them also pharmacologically active. So do things like go beyond blocking and inhibiting, but activate or otherwise tune the activity of receptors or targets that they bind. So that's the sort of major technical gap that we're trying to fill. And turns out that there are a lot of drug targets where, hey, if only we could more specifically activate or increase the activity of this, that would be a great way to treat this disease. And prior molecules, small molecules, peptides, just haven't been able to get the specificity to, um, you know, either make it through clinical trials, or once you do, there's still large segments of the population that can't benefit from them. And can go into detail about our major program, which is in obesity, and of course, the fact there on the ground being that large percentages, 20+% of people in real world end up not taking these or stop taking these obesity drugs, the GLP-1s and the various G drugs after the first year because of the gastrointestinal side effects, right? So, and that's because you just have these drugs that light up neurons all over the brain and body. So fast for the functional antibody selection technology. I'm getting there. I'm getting there. We got 45 minutes, so
We got time.
so the idea is that what if we can directly test huge numbers of antibodies directly for activity, directly for their ability to modulate or alter the activity of the things that they bind. If we could do that, not only could we find these really rare molecules, like that special key that just fits into that lock just right, that opens it up, right? Not only that, but can we now take advantage of the awesome large language models, protein-based large language models, and all the artificial intelligence tools around that, and actually learn from that data, right? And then can we actually get into a regime where we're we're not just trying to get lucky and find antibodies that do what we need to do, but to actually learn from some basic data and be able to extrapolate and ultimately design molecules that we're interested in, that had the function that we want. So that was the sort of underlying dream, underlying the FAST platform. It turned out to be working quite well. We've done this for a couple GPCR targets, G protein coupled receptor targets, notoriously difficult target class for activating antibodies. Just to put it in context, there's only, as far as we're aware, there's only been 9 GPCR targets that have ever been activated with antibodies, and we've done 2. So we're hot on the heels of getting some data on a couple more. It's a nice, apparently scalable program or platform that we can apply to other targets. And of course, biology being what it is, or maybe some problematic ones, but thus far it's been working well for us. Um, and and at a very high level, and then I'll stop talking so this can actually be a conversation rather than a monologue.
Hey,
Um,
I got
basically we have engineered cells, all those ologies you mentioned earlier. So that underlies the platform technology where we've engineered cells, specifically yeast cells, to not only make human target proteins of interest, but then individually test antibody variants for function against that target per cell. So that's one of the reasons why we're named Abalone. It's every cell tests one antibody alone, alone, right? Per cell. Yes.
got it. I got it. I was thinking it was just a play on seashells.
Yeah, co-founder was a Abalone diver as well, so we can go into that. But yes, so that's the basis of the platform, and I can go into more details and explain it in general terms in a bit. But essentially, we found a way or engineered a way to have cells individually test antibodies for function, measure that functional data, and then use that data to either identify or ultimately create molecules of interest.
Yeah. Thank you. Well done.
Thank you. Thank you. Well done. Yeah. When will this guy let me in? Yeah.
No, hey, I love hearing talk about their passions, and obviously this is working well for you. Like, you're onto something, and it's something that I see a lot of people talk about high throughput screening, right?
Yeah.
But for our audience are your peers, people in biopharma who are developing and manufacturing same kind of therapies, and so for them, and for me, because I'm obviously not a biochemist, I want to know, when you say functional screening, when I hear that, it makes sense. But if we dig in a little deeper, I'm curious what exactly it means to screen functionally. Like, how does that compare to traditional affinity screening? I'm curious if manufacturability is considered when you're looking through and screening these things. For example, expression yield, antibody stability, developability.
Mhm.
Yeah, I'm curious to hear your thoughts on that.
Yeah, yeah. Definitely have thoughts on that. So maybe I'll go into a little bit more detail and how the platform works. So
Without giving away too much secret sauce.
yeah, it's fine. I can talk about it in general terms. So yeast cells, of course, are very sort of basic eukaryotic cells, right? And they actually have a lot of conserved machinery between themselves and human cells and other mammalian cells for that matter, including targets like G protein coupled receptors and all the machinery that responds to and carries forth into the cell signals from GPCRs and elicits downstream biological effects, right? So what we have done is take advantage of that similarity, and also the sort of simplicity of the yeast cells and also the engineerability of the cells. So we can delete the sort of a few endogenous G protein coupled receptors. I should have said at the beginning, we're focused very much right now for antibodies for G protein coupled receptors. That's where we see just a big opportunity for the kind of molecules that we can find. And we can go into later about how this platform can be extendable to not only other target classes, but other drug modalities. But for now, GPCRs and antibodies,
Yeah.
we can delete yeast GPCRs in their genome, replace them with human GPCRs, or a human GPCR target of interest. Then that GPCR is expressed, transported to the cell membrane, and we have to make some mutations to the downstream machinery so that the GPCR can talk to the yeast GPCR signaling machinery. But we do that, and we make sure, we can, we test for activity. We typically focus on validated targets, right? So we can use known ligands or tool compounds or other compounds that there's published activity on. And we make sure that we see reasonably similar pharmacological properties in the yeast pathway. And then we can connect to that yeast signaling pathway to whatever readouts we want. The powerful thing here is actually to get out of the screening regime, right? So we can hook it up to GFP or luciferase or whatever outputs and do flow sorting, right? But when you connect them to survival markers, right? Some octotrophic marker or some antibiotic resistance marker, something of the like, then you get into the realm of selection, which is a lot more powerful, right? Because then you can essentially just let these things grow, right? In these cells, the same cells that have a functional target, we have a single copy of an antibody variant and express that, and it's transported outside of the cell. And the cool thing about yeast cells is that they're like these quasi-plant cells, right? So they have a cell wall, a mesh outside of it, which allows small molecules, peptides, ions, water, that sort of thing to go in and out, but not large proteins like antibodies or even heavy chain domains and look the types of antibody fragments that we're working with. So these cells, they make a target of interest, and they also make a single antibody variant, and they're both in this extracellular space. That antibody does not float out and go to another cell. It stays restricted to the same cell. It's sort of like a cis or autocrine-like signaling. It basically is like poking itself to see what it does, right?
Yeah.
And then if that antibody triggers the signaling cascade and we have it connected to a growth reporter, that cell multiplies and proliferates more, right? So now we've essentially engineered, animals on the Serengeti situation, right? Like rugby scrum / survival of the fittest, right?
Yeah.
So we're basically let nature tell us which ones are active, right? And this is, I think, a point of differentiation from a lot of the super awesome work that's out there based on, structural work, the alpha fold and all the derivatives thereof. I mean, it's amazing that we can predict binders with antibody binders with any accuracy and reproducibility and efficiency. I mean, it's like astounding, right?
Yeah.
When I grew up, protein folding problem was like one of the great unsolved problems, right? So it's like solved now, basically, right?
Heck of a-
Yeah, back in my day, we can go into I can a lot of uphill both ways, no shoes, yeah. But
to sort our antibody.
that's right. That's right. I mean, that's why I got out of crystallography, which is what I did my Ph.D. in. I was like, I saw the robots on the horizon. I'm like, all right, this is I can see myself being made obsolete very quickly here.
You forever.
But yeah, yeah, yeah. Fear is a great motivator.
You think I'm a writer? I can't. I can't even-
Yeah, I could be
You're already
talking
here
to
for my job.
I know I could actually be talking to some GPD avatar right now. I'm
Not even real.
There exactly. Yeah, I don't know. You're amazingly convincing. um, yeah. So again, the point of differentiation being that we're actually quite agnostic to the structure. We are right now at the next level incorporating structural information. I mean, it's out there, so we want to take advantage of it. But fundamentally, we have this naive search through the sequence function landscape, right? And and that allows us to, in an unbiased way, like not get caught up in these sort of like local minima or cul-de-sacs of something that binds very tightly, but that actually doesn't do anything. And that analogously extends to your prior question about other sort of wet lab based techniques, right? Where you're right, they typically look for high affinity binders. Makes sense. You want to bind something to have activity. But what those methods end up doing is they end up focusing antibodies on, immunogenic regions, and then you start driving towards higher and higher affinity interactions. which may not be the parts of the target you want to bind, actually adjust the structure in the way to give, a functional output that you want, right? So binding, of course, is important, but it's not um, tantamount to function, right? And in my pitch decks and slides, I usually have a picture of like two ways of grabbing scissors, right? You can grab them like really tightly by the blades, and it's a high affinity interaction, but it doesn't really help you, cut anything, right? But if you actually loosely bind scissors, but in the right way, and just the right places, then you actually have a functional interaction. In a similar way, we think about antibodies binding the targets of interest, particularly GPCRs. You have these like 40, 50,000 atoms that are jiggling around, highly dynamic. Um, Structure information is also generally static, so it's like it's a incomplete thing. It turns out to be much more difficult for GPCRs.
I just want to stop you and say that that was a fantastic analogy. It really put it in perspective with the
Yeah.
scissors.
Right. Yeah.
Definitely be using that if I ever
Yeah. Yeah.
next time I'm
Please.
talking to people about the difference between affinity and functional
Yeah.
antibodies. There you
Exactly.
go. I think that's
Yeah.
perfect.
Yeah.
Um, Now I wanted to move into you briefly alluded to your pipeline and indications and strategy. I do want to talk about that, and I also want to ask how you're preparing those for
Yeah. Of
IND.
course.
But I did before we get off the topic of fast and the platform. I was reading about how the company was born out of this aha moment, and I'm curious if it's something you already discussed, or if it's if there's if you could walk us through that moment that you were like, oh my goodness, we could do something, we could do this differently than anybody's doing it right now, and in way better.
Yeah. Yeah, well, so, my co-founder, and a very good friend, Gustavo Pesce and I, our postdoctoral work was with, Sidney Brenner and Roger Brands at, one of the first systems biology institutes called the Molecular Sciences Institute. And oft unappreciated, fact that this is actually the birthplace of the synthetic biology movement as we know it. We were all there. I mean, there are the luminaries in the fields who went on and to create that field, or be the sort of OGs in the field, like Drew Endy, Rob Carlson, all those guys. I mean, we were talking about whether it should be called synthetic biology or intentional biology, and like not quite sure. Like it all sort of came together there. I don't think many people appreciate that, but it's sort of cool. And I think it was because it it was an institute that really put together it was the first real interdisciplinary place that I worked, right? I mean, we had a particle physicist, we had a straight up hardcore computer scientist, we had biologists of all different shapes and flavors. And I think that's where, the magic always happens at these intersections, right?
Yeah.
And similarly for our technology, we had spent many years studying how yeast use a prototypic GPCR signaling pathway to process information from an external environment and convert that into cellular behavior, right? And I was on more of the basic biology end, so it was like, using Shannon information theory to like try to understand what's the bit rate of, the signaling pathway and things like that, and what we can learn about genetic control of those elements or those properties. Pretty sort of basic biological research. Um, but I'm get there's so we had all this underpinning on this very interesting biological system, and we wanted to for Abalone to work on something that had in terms of a company, right? Something that had a reasonable brackets on the sort of risk profile, particularly as you go downstream. And to say one word, I had also done a algae biofuel company before, which is like one end of the spectrum on downstream risk, which is like a ton, because no one knows, are you going to make, 100 billion barrels of algae oil, per month or whatever the metric
Yeah.
is. But with antibodies, it's like, okay, yeah, this is a place where we can actually make a difference. If we can find the antibodies that we that have the properties that we want, that have the functional activity that we want, then, how you make them, how you develop them, how you market them, sell them, all that is like, at least there's a roadmap. So for us, the aha moment was this observation that most antibodies are sort of blockers. Talking to our friends and colleagues who were in pharma at the time, yeah, they're saying GPCRs are definitely this interesting white space, right? Because they're one of the most validated target classes out there, but yet at the same time, one of the most under harvested, right? I mean, what is it like roughly a third of drugs target GPCRs, but only a quarter of them have been drugged, right? It's like, is this really interesting asymmetry in terms of risk profile, right? So we looked a little further into it and we're like, oh my God, people have already used yeast to do small molecule drug discovery. But one thing that we got really beaten into us, both individually in our postdoctoral work and graduate work, and then at this institute, was the awesome power of genetics, right? You know, the Ira Hershkowitz UCSF mantra, and the idea that we could, if we can use sort of genetic strategies and principles, like selection, to interrogate properties of interest, like functionality of an antibody against a target, that would allow us to find really rare variants in a sea of variants in a huge initial population. Um, the thing that no one had put together, and we were just actually like super surprised that no one had done this before, was to combine yeast that make a human target of interest and are responsive to a human target, and combine that with an antibody display. Oh, yeah. The other part is, of course, yeast surface display has been used for, Adamabs and the likes, right? I've been doing that for a long time too, right? But no one had really put those two together, right? So antibody displays on the outside of the cell, and you're using the cell as like a living bead to just sort of hold onto the DNA that encodes that antibody, that you can bind it to things and wash it away, right? And look for tighter binders, if you do affinity maturation and the like. So what we did is we did that little tweak. We're like, hey, what if we presented it on the same cell, like in a cis-like fashion, that would interrogate itself for function, right? Yeah, so we did some POC experiments with, some well-characterized receptors and ligands, and yeah, I mean, the cartoon like worked,
Yeah.
it's been many years of detail, mashing and making it work well and robustly and repeatably. But that was the genesis. That was the sort of aha moment. And yeah, we couldn't believe that no one had put those two together. And sure enough, yeah, we were able to get that to some claims issued in numerous jurisdictions, right?
Thank you for sharing that. That's great.
Yeah,
Um, I'd be remiss not to talk about the pipeline of biologics you guys are working on, the
yeah,
antibodies. And I saw, on your website, you guys are across the board, you're working in metabolism, inflammation, and oncology. Those are the big buckets, I think.
yeah.
I'm curious if you could share whereabouts your candidates are in progress, and how you
Mhm.
are. I'm gonna try to shoehorn a couple questions into one here.
No, no, go for it.
I'm curious how you're preparing these candidates to move into the clinic, because you guys are all preclinical, correct?
Yeah,
Um, so I'm curious how you're preparing for IND, these assets. If you could share a little bit of your philosophy is in terms of strategy for treatment. I know I talked before about GCPR being the main targeting, and
right.
I was curious if how the company's planning to transition into GMP production, whether you're planning to partner up for that, bring that in-house, or is that down the road and you're worrying about this most immediate issue that's right in front.
Yeah, yeah. Um, that's a great multi-layered question, and I'll
Sorry,
try to
those are
chip away. No, 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 it's great. It's actually a great, and there's a great line to that. I just want to make sure I don't forget any key elements. So please take notes, and we'll go back to stuff if I don't address something of interest. Um, our pipeline. Look, GPCRs are everywhere. I mean, the fact that we're saying metabolism, inflammation, and oncology doesn't really narrow it down all that much. If you consider those three mega buckets. The idea is that we're, of course, first narrowing down on targets, first and foremost, requiring the sort of specific activation or positive modulation. We feel that, competitively, um, there are other technologies for finding inhibitors or blockers, even of GPCRs. As difficult as they are, that's like not. I mean, we can do that. We can look for things that turn down activity too. But the real differentiation is in the activation. So we focus on targets that require that. Um, as a small company too, of course, we're thinking about targets that are appropriate for partnering and at what stage of partnering and how much we want to take it downstream. Do we want to raise the money to do the clinical development? How far do we do the clinical development? And the real answer, of course, is being opportunistic at all times and flexible. Things can change on a dime, especially these days, and the sort of big external factors. But I think there are also some fundamentals that provide a North Star for us, right? I mean, the metabolic space, we do see a interesting white space opportunity for specificity or avoiding one or both of the major side effects or drawbacks of the existing drugs. Excuse me, getting over a bit of a cold here, so it's a little raw. So that would be the GI side effects, which I mentioned earlier, and then also the fact that the muscle loss or the weight loss has a pretty heavy component in muscle loss as well, right? So if you could find targets that could address one or both of those issues, um, the market's been proven, the market demand has been proven.
Billionaire.
Yeah, exactly. It's not just that. I mean, that's great. That's what our investors want to hear, of course.
Of course.
But I mean, there's a great article, I think is by Omri over at NFX, who talked about this idea that these are really like the GLP-1 drugs are like sort of the first essentially proven anti-aging drugs, right? Or life extension drugs. If you think about it in that way, it's absolutely true. It's like, it's finally, it's like so hard to get into preventative mindset, but that's essentially what it's doing, right? It's like by treating obesity, you're treating all the downstream consequences of obesity. Um, and so it's really uh, a high impact um, um, class of molecules, right? It's yeah, I mean it does happen to be a way to make a ton of money, but I mean you can really impact a whole a slew of diseases with that. So it's really appealing in that way. And the fact that we have a very clear biological thesis on how we want to attack it, right? And that fundamentally is that if you can get specificity for specific target or specific target subtypes that partition in the right places, or we can find cell surface markers that allow you to generate molecules with the antibodies. You can do bispecifics, right? So you can have one arm of an antibody sort of localizing the activity at the other arm of the antibody, right? But generally speaking, either target or cell or tissue specificity is the way to separate the beneficial effects from the side effects. If you can identify that biology and have a mechanism for that, that's what we've done. So we've identified targets that sort of fall into that general mode of action. Yeah. So in terms of where we're at, we have one molecule in the metabolic space that's at a development candidate stage. This one has been, we're looking at it in a liver fibrosis and MASH indication. Got some good animal data there. And like I said, we have a development candidate selected and some backups, and we're just starting, the cell line work right now. In terms of how we're doing it, I mean, these days, at least I think there's a sort of a bimodal distribution of funding. You got the billion-dollar seed rounds, and then you have the not billion-dollar seed
Yeah.
rounds, right? So,
Yeah.
for the vast majority of us who are in that second bucket, staying lean, using consultants, the CROs, that type of thing is definitely our strategy, and probably will be for the near term. If there's, of course, ever an opportunity to hire exactly the right person, the right CMO, all that to drive these things to the next, value inflection point, as they say, of course, we consider that. Right now, we're early enough that we can, we're making the initial forays into clinical development, or in this case, preclinical development, in a sort of lean fashion. Some of the early, so now, yeah, so we've had this molecule, it's proved out the platform, it's manufacturable, it seems to be you have all the right properties. One thing I didn't mention before to address one of your earlier questions is um, sort of developability, expression ability, and all that. One of the nice things too about our platform is that when a cell is making an antibody that causes it to grow, it's an integrated measure of a number of things. It's an integrated measure of how well, frankly, the target gets to the cell membrane, but also how well the antibody is expressed and transported to the outside, right? So that in and of itself is a sort of filter against sort of crappy molecules that, you know, precipitate or whatever, right? So there's a sort of a nice bonus in the platform that it already sort of has at least some cut on antibodies that can express well in a eukaryotic system. And I guess, as I understand it, there are some large pharma that are shifting towards like picky expression and other modes of expression for antibodies and biologics. So the fact that we have at least one yeast readout is helpful. Um, yeah, and then our other programs, these are programs that we've shored up and developed our biological theses on relatively recently. So we're in discovery right now on the other sort of follow-up programs, and these are in the more directly attack some of the problems in the obesity, whether it's the side effects or the muscle loss, muscle wasting.
It's great. Thank you.
Yeah.
Um, kind of going all over the place. This question is that.
Yeah, I hope this is I hope I'm
No, no, you're
coherently answering or at least laying down a narrative thread that at the very least I hope is amusing to people and not
you're
informative.
doing a great job, terrific job. I just keep getting new questions popping in my mind that I'm like, oh, I need to go back to that. So,
Lay it on.
folks, apologize if you're having trouble keeping up here, but I really wanted to take it back to the AI component. I mean, obviously,
Yeah.
built this company on the platform, which is AI is part and parcel with it, right? And I know there's no shortage of discourse on AI. We don't even
Yeah.
need to really go down that route. But I was curious. We will go down that route a little bit, but I just want to know what your thoughts on, if you could share them with your peers in the audience here. What would your advice be when it comes to adopting a new kind of technology, whether it be high throughput functional screening like AI or even just general guidance when it comes to parsing through all this AI noise to find
Yeah.
out what is valuable for biopharma, what can be beneficial to you, and how can you design if you're in the beginning, how can you design the company to really embrace the changing tides of technology? But if you're pretty well into it, how can you be what's just the philosophy you should carry with you to when it comes to technology adoption and weaving that into your operations?
Yeah, that's such a great question, and I should appropriately put some dotted lines or brackets around where I think I can be most informed. I'm generally in the sort of earlier stage, thinking about molecules and structures and activities, right? So I have a layman's understanding of, how to properly use AI in clinical data, you know, mining and all that stuff, right? So just to appropriately bracket my at least self-graded expertise. Um, so I think look, I did some early AI work on protein structure prediction. This was in the early '90s. You can believe that. Um, Back at Berkeley, this is sort of there's a brief blip in this sort of crossover of AI and bioinformatics. And it's like, hey, there's all this sequence information. Maybe we can, extrapolate to, you know, novel properties or new sequences that give properties that you want, right? Um, and we quickly noted that this is for protein secondary structure prediction to just get to the details for a moment. Um, but there were 400 structures in the PDB at the time. I think when AlphaFold 2 came out, there were 177,000, right? So, and they're all globular. I often joke that like there are 400 structures and 350 were lysozyme. Like it was just a very short or small pool, and not very diverse pool. and transformer-based technology, the animal side hadn't come out yet, right? So is quite limited. It's very much like this much more simplistic, approximation machine, right? So the data just wasn't there. So I have this like deep rooted um, sort of prejudice to on the data generation side, the novel data side of things, right? As a person who's technologically inclined, is also aligned with my interest in developing new technologies as well, right? But I think that's where it is most. I mean, like most things, right? That's where you're going to be most differentiated and have the most impact, right? I mean, to say you're using AI in your company right now, it's almost like saying I use PCR in my company. It's like, well, duh. I mean, everyone uses it,
Yeah.
right? It's like, it's not differentiating. Um, I think if you, there are companies who are developing legit novel amplification techniques. So that would not be a PCR company. It'd be a new next gen PCR, whatever, right? And similarly with, or analogously with AI. But I think, um, am I going to compete with, Google, Meta, engineers on, and their various, progeny and the people who've left those firms to start their own AI companies? Not on the ML innovation side of things. So it's mostly going to be about the data.
Yeah.
So that's what we've focused on, and that's what we have, and that's why I think. I mean, just operationally, we've demonstrated that that's where the difference is. I mean, we've been able to find over 20% of the GPCR antibody agonists, right? You know, um, 2 out of 9. So these are not large statistics, but I can do the division and get a percentage, right? Yeah. So I think in terms of how, you know, um, where else to put that, it's also to think about where novel data is important, right? Based on my structural biology and sort of protein biochemistry, um, training, like in my mind, I can see how we can go from sequence to structure with ML now. We can go from structure to binding in some cases, right? But I still see that there is a large gap between what types of interactions confer different types of activities, particularly for large dynamic proteins, right? So that's the place where I think there's a data gap that ML can't extrapolate right now, or very efficient. I mean, there are a couple isolated examples, but they're typically, based on a peptide that's bound, so you can sort of like get really close with an antibody, right? Still amazing work, but it's like, what happens if you don't have a structure of the target, or what happens if like, for example, many of the targets we're looking at, um, they are small molecule binders, right? So we want to take the advantages of antibodies there, but the antibody is not going to go in and sort of mimic the direct orthosteric binding site of a small molecule, right? So I mean, what are you basing your AI on, right? You have to like, there's hope that once we start measuring enough data, that we'll see common structures and modes of activation in GPCRs that we can then work backwards to say, okay, what molecules could bind to give that effect, right? But I don't think the data's there yet. Operationally, again, that's sort of shown by the fact that there are just very few GPCR antibody agonists that have been certainly intentionally discovered or designed. So, yeah, I think it's just thinking very carefully about the overall process and where are the gaps. And like I said, in my mind, it's really focusing on generating some differentiated data if you wanted to build a company that has a moat of any sort.
Yeah. Well, I hope Abalone stands as a testament to what can be achieved when we do team up with the robots and give in
That's right.
to our robotic overlords.
I welcome our robot overlords. Yes.
Right. Well, Rich, I don't want to hold you for too long, but I did want to ask you this question that is kind of the central guiding question to the program and that I pose to every guest, and that is simply how can we better biopharma? In your opinion, what's the single greatest thing we can do to push the industry forward? Whether that's
Yeah. 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 Again, my biases are sort of on the earlier um, part of the funnel. right? Like if we can find or design better molecules going into the funnel that have some hope of shortcutting some of the later processes that can accelerate drugs and get them to patients faster, right? That's the guiding the North Star for all of us, right? Um, that's where our expertise is at. I do think that with the promise of ML and AI, that if we can magically um, combine and analyze data across the whole sort of development chain from discovery to patient, that there are some shortcuts that can be identified, some really accelerating shortcuts. Um, so yeah, I think that's on the science and technical side, right? But of course, there's always like the policy side, right? And like what and the financial side as a sort of extension of that, right? So like, what are not just the drivers and the pushers, but what's pulling things out, right? So this is like way above my pay grade, but thinking more broadly in terms of how do you set up the right financial incentives and structures, regulatory structures, yeah, pricing structures to make sure that all of the parties that play in this process that are essential, and you can argue whether PBMs are, part essential in that part of the process, the overall environment. But yeah, well, how to align everyone to make it as productive as possible. Yeah, I mean, those are the things again I think about when I read like the more the sort of policy side of things on fears or endpoints or whatever, right? Or the um, you know, as we started off in the sort of pre-chat before the recording, that there are of course also things that are, as they say, outside of your circle of control, right? So I don't spend too much time thinking about that. But yeah, I mean, if I could wave a magic wand, that would be certainly a part of it, right? There's got to be a better way to set up the system. It can't be the most expensive and like the least have the lowest outcomes, right? That's like there's something wrong, right? Um,
Yeah.
yeah. So it's yeah. If there's some magic wand, that's sort of what I would address. But how? I don't know. I'm just focused on making better molecules. You know, do that my part there.
Yeah, we're all just trying to do our part. Well, Rich, thank you so much for joining me on Better Biopharma today. It's been great talking to you and to our audience out there. Thank you so much for tuning in. You can find more about uh, biologics development and manufacturing at Bioprocess Online. We'll see you next time.